you can probably start anytime.
Okay. Good morning. Thanks for joining us for today's seminar series. Today, what we thought we would do would be to share some information about two incredibly useful resources that the Cancer Center supports. One is the Genome Engineering Shared Resource, and the other is the Mouse Genetics Laboratory, and they kind of work hand in hand. And so the way that we've got it structured is that Brian Ruis, who's in charge of GESR, the Genome Engineering Shared Resource, will describe what he does in his resource, and then Dr. Yun Yu will describe what goes on in the Mouse Genetics Laboratory. And then we'll have a discussion after each of the segments, and so you can ask them questions and try to understand how best to use the resources that they have. So with that, let's go ahead and start the presentations.
Okay, yeah, so I'll go first then, and let's see. Yes, can you all see my presentation?
Yes.
Look good? Okay, so hi, my name is Brian Ruis. I'm the coordinator for the, as Chris said, I'm the coordinator for the Cancer Center's Masonic, the Genome Engineering Shared Resource, or GESR or GESR, whichever one you want to call it. The GESR is a P30 Cancer Center support grant supported shared resource. We established this in the fall of 2015 to provide at cost genome editing services to the cancer research community. To date, we're able to produce a total of 75 mutant cell lines, as well as collaborate with the MGL on a total of 30 mutant mouse projects at this point. And the shared resource is located in the Molecular Cell Biology building on Washington Avenue, and our website is shown at the bottom of the screen here. So again, like I said, we've developed 75 mutant cell lines, and most of those cell lines are human cancer cell lines. On this list is all the, encompasses all the cell lines that we've actually worked on over the years. So not only human cancer cell lines, but also some primary cell lines, along with some non-human mammalian species such as dog, mouse, and rat. Along with this, with the success of being able to generate so many cell lines, we've also been able to support peer-reviewed publications. And we have, this is the total number, at this point, we have 5 publications that the shared resource is supported. And we have currently 2 more publications in press that will be coming out shortly. So CRISPR-Cas9 is the method that we use in the shared resource, and CRISPR-Cas9 has been around for nearly a decade now for a lot of different uses, including human disease therapy and new drug screens. But again, what the shared resource focuses on is making cancer cell lines, modified cancer cell lines, and also modified animal models. What we provide is CRISPR-Cas9 editing for knockout of essential genes, and I'm trying to move my knockout of non-essential genes, conditional knockout of essential genes, epitope tagging, single amino acid knock-ins, single SNP knock-ins, and multi-allelic knockouts. So the way we do this again is CRISPR-Cas9. Cas9 nuclease can be guided to nearly any locus within the human genome, and if the Cas9 along with the guide RNA will produce a double-strand break. This double-strand break, if it's repaired by the non-homologous end joining pathway, will produce insertions and deletions at the cut site due to its inaccurate repair events that happen. Those repair events, are commonly, insertions and deletions are commonly referred to as indels. This is the method that we use for doing non-essential gene knockouts. We use Cas9 mRNA along with the synthetic guide RNA. Alternatively, conditional knockouts and of essential genes, knock-ins and gene tagging can be done using, again, Cas9 mRNA along with the guide RNA. But this time, homologous recombination can be used to incorporate new sequence with a donor DNA. And this donor DNA can be provided as either a single-stranded oligo, double-stranded DNA plasmid, or an AAV viral donor. So the way that the workflow for the GESR to do knockouts is to transfect target cells with Cas9 mRNA along with the synthetic guide RNA using a neon electroporator. We've been doing this for pretty much since 2015. We then expand the cells for Cas9 cutting analysis by TIDE. TIDE is a computer web tool that was developed several years ago, and it stands for Tracking of Indels by Decomposition. Alternatively, Synthigo, a company that provides synthetic chemically modified guide RNAs that we use, also developed a web tool called ICE, which stands for Inference of CRISPR Editing. I will go over just the TIDE analysis for this example. So the way this works is the web tool is designed to rapidly assess genome editing by CRISPR-Cas9 cutting activity at the target locus. An amplicon is needed for this for the modified cells, either a polyclonal or monoclonal cells. Along with that, a reference amplicon is needed for the non-targeted cells. And ideally, an amplicon of about 400 to 600 base pairs is required for this. So the way that this works is after PCR amplification, chromatograms are produced. These chromatograms can be plugged into the web tool along with the guide RNA sequence to denote where the cut site is. The web tool will then compare the reference sequence, which is shown here in black, compared to the edited sequence. And at the cut site, the sequences diverge, and the web tool then uses that difference and determine accurate, or with quite a bit of accuracy, predicts the amount of either one base pair insertion in this case, or a one base pair deletion along with some unedited sequence. So once the highly edited population is determined to be edited, we then subclone these cells into 96-well plates for single colony expansion, and determine looking for knockouts at that point. We expand the colonies onto 24-well plates, or doing direct lysis from 96-well plates. And then once the clones are collected, we collect genomic DNA from single individual clones, again, PCR amplified as before, sequence the resulting amplicons, and then use TIDE analysis to determine the indels within each clone and expand them for out-of-frame indels. And then downstream Westerns can be done for looking for knockouts. So in addition to using CRISPR-Cas9 in the traditional sense for making either knockouts or knock-ins of sequences, two other new editing techniques have been described, and we have the potential to use these for future processes or projects, including for base editing and prime editing. So base editing is a good technique because of the fact that, so of the 32,000 pathogenic human SNPs within all the diseases for genetic diseases, more than half of them are caused by transition mutations. And each of these transition mutations can be corrected either with a cytidine base editor or the adenosine base editor. The way it works is Cas9 been tethered to a cytidine deaminase or an adenosine deaminase, and upon binding to a target sequence, nucleotides within a certain range of from the cut site or from where the PAM sequence is, will be amenable to base editing, where cytidines can be converted to uridines, which will then result in a C2 to TA base conversion. Alternatively, adenosine base editor will convert an adenosine to an inosine, resulting in an AT to GC base conversion. So this is a potential tool for us to use in the future. We haven't done any projects with this to date, but I anticipate using this in the future. In addition, another new tool was developed in David Liu's lab in Harvard, where a Cas9 is tethered to a reverse transcriptase, such that when you add on to nucleotides to the end of the guide RNA, extend the guide RNA 3' end, you can allow for a primer binding site to be formed on the non-target strand. And if you incorporate nucleotides within this sequence that are, you can insert nucleotides at that site to allow for incorporation of mutations. And this is good for all, so base editing was good for transition mutations, but sometimes the transition mutations are not within that window of opportunity to be able to be edited. So prime editing can edit at those non-base editable transition mutations, as well as in incorporating transversion mutations in deletions and also insertions to allow for other corrections of pathogenic genetic variants. So the rates that the GESR, we have several types of services that we provide. So first we have ones that are we call non-phase based. And this would include, these are the table of the fiscal year 2022 rates. And this includes consulting and cell culture time. We charge this in a per 15-minute basis. And I'm on this table, I'm including cancer center member rates, non-cancer center member rates, and also the external rate. So we also have guide RNA design, guide RNA editing validation, and we do this in the target cells that we're working with, and then also HDR donor vector construction. In addition to the non-phase based, we also have phase based, which are including the generation of mutant cell lines. And the way we build this out, we have 3 phases. The first phase, which we bill at 1/4 of the cost of the project, will be billed upon completion of each of the phases. So the first phase would be the project design and ordering of the editing reagents. Phase 2 will include initial editing being performed and the editing efficiency being determined. And then if everything looks good, subcloning will be initiated at that point. Usually the way that the projects work out, we usually end up building both 1 and 2 at the same time, just because of the amount of time it takes to do the first one's not that long. And then finally, the phase 3 is completing the project where correctly edited clones have been identified and provided to the individual lab if possible. These are the rates for fiscal 2022 rates, again, with the internal cancer center, non-cancer center, and the external rates shown for knockout of non-essential genes, conditional knockouts, it's epitope tagging, single amino acid knock-ins, single SNP knock-ins, multi-allelic knockouts, and we have a price for CRISPR-Cas9 whole genome knockout library screens. We have not done a knockout screen for the GESR to date, but I have myself, in the capacity of working in the Hendrickson lab, have performed at least 3 of these genetic screens, knockout screens myself with success. So I have experience in doing this, and we can certainly talk if anybody wants to do that kind of thing. So with the collaboration with the MGL, we have certain parts of that workflow that are either GESR responsibilities, MGL responsibilities, and possibly user responsibilities. And so the way that the workflow goes for that, we usually would like to meet with the clients first to discuss the project. Once the project has been determined, or the target is and what we're doing, I will then go through guide RNA design and validation. Validation is done usually within NIH 323 cells. And this takes several days. We then, depending on whether or not it's a knock-in, if it's a simple knockout, we just do just a guide RNA. But if we end up having to do an HR mediated event, we usually use an AAV donor. This will take several weeks to design and order and design and generate the donor. once the donor arrives, then it's we have packaged it into AAV6, usually through a company called Signigen. And then once those have arrived, we then prepare the reagents for injecting it the MGL. And then Yun will discuss what he can provide later on with these. And then finally, once the pups are weaned, toes are clipped, genotyping can take place. Either the GESR does offer genotyping services, but sometimes individual labs may choose to do that as well. And then finally, the requests for the genome engineering shared resource can be put through ILAB. So we have the top part of this page shows the actual website link where you can sign in or register for ILAB. And all the billing and all the updates and stuff are put through ILAB. And finally, I'd like to thank everyone for attending today. I'd like to thank the co-directors, Eric and Brandon, and my technicians, Summer and Chris, as well as Tim and Yun of the MGL, Chris for organizing this talk, and then all the members responsible for establishment of the GESR in the first place. And I'd like to answer any questions you might have.
That was fantastic, Brian. Thanks very much. Are there questions from the attendees or comments from people that have used the GESR facility? Well, I'll start off with a question then, Brian.
Sure.
What is the major limitation, or what is the major problem that people have encountered when trying to modify genes and using some of the techniques that you've described?
I think one of the main problems, I guess, is the number of chromosomes that some of the cell lines so a lot of the cell lines we use are cancer cell lines, and they're aneuploid. And so sometimes trying to make knock-ins or knockouts are usually pretty easy, even when you end up having an aneuploid cell line. But when you end up having a cell line and you're trying to make a homozygous mutant of incorporate a SNP or some sequence that requires it to be a homozygous cell line, it can be difficult and challenging to try to get all copies of it in. So that's probably the most difficult aspect of it. Also, I guess, depending on what the target site is. So some targets are easier to work with than others because of the GC content of the genome at the cut site. So if we end up trying to incorporate a single nucleotide polymorphism or something like that at a specific site, or it can be difficult at that point. Yeah.
Okay. Dr. Yi has added a question to the chat, and he asks, do we have a policy regarding sharing of cell lines that are made in GESR? And if other members want to use a cell line made for another lab, how does that work?
We don't have a policy. We've never had that request happen. No, we don't have a policy at this point. That's a great question. I don't think there's any. I don't know. We do store our cell lines. So every time we make a cell line for a client, we do store frozen stocks of them before we give them to the lab. So we try to make sure that we've validated it twice, so we validate that we found the clone initially, then we go through a process of validating that we still have the clone, and then and that it still is coming out the same way it did, that the results were the same the first time that we identified it. Then we make frozen stocks, and then once the frozen stocks are available, then we can contact the client, and depending on how many clones we have available for them, they'll get all the clones that we've been able to identify. Usually we like to try to do more than one mutant cell line. So if we're making a knockout, we like to give them at least 2 isogenic clones from that knockout attempt. Just for publication purposes. A lot of publications like to see experiments done with multiple clones. And maybe not the entire publication, but at least initially.
Do you then, go ahead, I'm sorry.
Oh, no, I was gonna say, but for policy for sharing between labs, we've never had that request done yet. So That's a good question though.
Okay,
Tim Holmstrom has his hand raised, so I'll unmute him.
thank you.
Oh, I don't really have a question right now. We just have a formatting issue after Yoon gets done talking. We have a few users that would like to talk, so
Oh, okay.
good for now. Thank you.
There was another question, Brian. Do you have any experience with iPSC modifications? So induced pluripotent stem cells.
I do a little bit. So I actually have a project right now. I'm working with another lab to have them do the iPSC work on their end. So I don't have, so the GESR itself does not have the capacity to work with iPSCs at this point. So we don't have the manpower or the experience working with them. So with these collaborations, I've been asked to do the project design and ordering reagents and testing them in other cell lines. And then those can be then given to the labs that have their experts doing it on their end. So we have not been able to do anything like that. That would be a great addition to our toolbox if we could do that in the future. Yes.
Okay. And then one additional question: have you done large-scale chromosome engineering, like 10 to 500 kb deletions or insertions?
I have not done something that big. No. I think the most we ever did was we were trying to incorporate mutations within a target region, and we had to cut out 8 kb, I think it was, but we never got the incorporation of the nucleotides where we were able to get the deletion, of course, but we haven't done really large deletions.
Okay.
Certainly possible to try though.
And if there are any people that are watching this video that have used GESR, if you could just chime in and just share your experiences with the resource.
Hey guys, it's Carol. Can you hear me?
Yep.
Yes.
I've had great experiences. One thing that we're doing, so we do have challenges because the steroid receptors we work on are really GC rich in the N-terminus. But despite that, they were able to make knock-ins of phosphomutant and phosphomimics in the past for us for glucocorticoid receptors and progesterone receptors and stuff like that. So that's been really cool. But what's been really cool lately that we're doing is, so usually we knock out the gene of interest and then re-express whatever post-translational modification mutants. But what's really cool is there's one gene for the progesterone receptor, and it has alternate internal translational start sites. And so what we're doing now is knocking out that ATG to make either the PRA or PRB isoforms instead of knocking out the PR and putting back in PRA or PRB isoforms. We're just making that ATG go away so we can have only PRA or only PRB. And so that's going to be really cool because it's going to be the endogenous promoter driving the expression of one or the other isoforms. And so I think that we can do kind of innovative things like no one's ever done that before. And it might be really different when it has its real location in the genome. So even though we don't have those yet, they're being screened right now. And we also recently made knock-in mice so that we can knock in the phosphomimic or the phosphomutant PR into the PGR locus. And those mice are also being screened. So I'm really excited about it, and everyone has been really helpful. It's been really much easier than I thought it would be, and it's been much faster than I thought it would be. So I'm really happy.
Thanks, Carol. That's great.
Thanks.
Eric?
Yeah, can you hear me?
Yes.
Yeah, I just want to sort of reiterate, you asked about people who've used GESR. I'm the head of it, so I actually haven't used it, but I think Brian can give you the numbers. But over the course of 4 to 5 years, we've had, I don't know, 75 to 100 successful clients. These are knock-ins, knockouts, alterations, the more sort of esoteric types of constructions that Carol just described, where trying to knock out internal ATGs or make internal ATGs. Brian and GESR have pretty much done it all. So we have at least 75 to 100, I would assume, happy customers out there. If it's, a reasonable project, it certainly can be done.
That's great, Eric. And I want to ask you and Brian this, going back to Doug's question about a policy regarding sharing of cell lines. If it's NIH funded, right, we're supposed to have data and resource sharing obligations, but that's on the PI, not on your facility. Isn't that correct?
That is absolutely, from a top-down point of view, if you want my opinion, we would never allow that. If you called up and said, could I have Bob's cell line, I would never give it to you. That's up to Bob to give to you. We make the cell lines, we can document every single step if that needs to be documented by NIH. But at the end of the day, the cell line belongs to the PI who paid for it. And they're now under their own sort of moral, ethical, NIH-driven edicts to, disperse those cell lines. But we would never do that without authorization from the PI.
Along those lines, though, you have a repository of all of these lines, right?
We do.
You do. Okay.
We do. They are all there, but we would only give them out to somebody other than the PI with the PI's express permission.
Yeah. Okay. And Doug just chimed in saying, I think that policy is right about sharing. I'm guessing most PIs would be happy to share, which we should. Yeah. But it's not your responsibility. Okay. That's fantastic. Brian, thanks very much. This was every time I hear you talk about this, I'm just blown away by the potential. And with Carol's comments, that's just fantastic. Looking at different splice variants in the context, in the the native in situ genomic context, that's phenomenal.
Yeah.
So thanks very much.
Thank you.
Okay, we're going to switch and then hear from Dr. Yun Yu about the MGL, which is located in CCRB. And Dr. Yu is he's unique. He's a unicorn. He's been phenomenal resource that we've been able to collaborate with. So Dr. Yu, please tell us what's going on in MGL.
Yeah, thank you. Thank you for the participation of this seminar. My name is Yang Yu, and I'm the MGL coordinator. And this morning, I had some glitches in my computer, I think. So Brian will make a slideshow because the issues with the computers, something not quite right.
Right. Okay. Just a second.
Yeah.
Yeah. Okay. That work?
Is that working?
Yep.
Okay. Thank you. Let's start next. Okay. Yeah. So mouse genetic lab basically function as transgenic facility for the university. I make transgenic mice, depending on what kind of mutation you want to make, maybe the simple gene knockout, maybe point mutations, conditional knockout, or maybe knock-in. Conditional knockout is part of the knock-in actually. Nowadays, we preferred method is probably by the CRISPR. Traditionally, you can also make transgenic by just inject DNA into the mouse zygote. That's a traditional transgenic mice. Few people asking for that, But I did in the past 6 years. I have 3 or 4 projects, but that's it. Yeah. So mostly I focus in the CRISPR. For CRISPR project, as Brian just mentioned, we collaborate together. The best approach, I think, once you have some project in mind, and let's have a meeting for MGL and for GESR, the genome engineering shared resource, and your lab. So we have 3-party meeting. You kind of specify your project aim and your ideas, and Brian and me will talk what we can do for you. So we have a understanding each other. And That's, I think, it's the best approach. Have a meeting first. Then you can we can schedule more detail. You know, some of the lab has a lot of experience with the CRISPR project, and in that case, they maybe prefer they design themselves, prepare the reagent themselves. That's okay for me too. So the MGL can sort of work with lab directly. So that depends on what you have. Then we can make the transgenic mice for you. After you receive the mice, I also like to remind you, we can do some consultation after you have, let's say you have problem with breeding your mice, you have maybe the genotype issues, you can talk to Brian or talk to me, you know, we can even have a Zoom meeting, let's say. These days it's relatively simple. So that's regarding the CRISPR approach for whatever mutation you want. Next slide, Brian. Yes, so some current limitation for CRISPR approach. So as I mentioned, for the simple gene knockout, point mutations, conditional knockout, or any knock-in project, if the size less than 4.5 kb, the CRISPR approach is very reliable. This was true, especially in the last year. In the early days, we have issue with conditional knockout. That's because of the LOXP sequence character caused that issue, I think. Also, people rely on the first conditional knockout paper, and that paper turns out has some issue, I think. But after we use the AAV virus combined with CRISPR approach, as mentioned by Brian, the result is much more reliable. So far, it's 100% successful for all, for all the project we use that CRISPR combined with AAV virus. That project, so far, all of them are success and have the mice. That's where the limitation comes from. That's 4.5 kb. That's the size AAV virus can accommodate. Larger than that, we can do that, still do the knock-in, use other approach, basically the plasmid DNA approach. But the efficiency to get the correct knock-in are much lower. As fragment increase, the successful rate is dropped very dramatically. Like I had a project, I think, try to knock in 11 kb or 10 kb fragment, and that project didn't work. We tried several slightly different approach, and it's not working. Yeah. Another concern for CRISPR for making transgenic mice, you have heard a lot about the off-targeting effect used CRISPR. It's not a high concern in the mice perspective. You can always breed them away. So next. So besides the new CRISPR approach, traditionally the other way is use the ESLs. The ESL can do whatever the CRISPR approach can do now. You can make simple gene knockout, conditional knockout, point mutation, knock-in, and that way knock-in usually is not limited by size, but still size is the issue. Once the size, knock-in size is increased to, let's say 10 kb, whatever, you usually get a lower efficiency. The good thing for ESLs, you can screening ESLs, make sure you get that mutation at ESL level. And once you have ESLs in a good quality, we can basically inject the ESL into the blasts, as you can see, and then make chimeric mice. You know, for the unique issue of the ESLs, you have to breed chimera, and they have to go germline, as you probably know. And if fail to go germline, then you have to start over. So the ESL quality, targeted ESL quality, is vital. So the ESL source, the source of the ESLs, some lab has their own. Maybe they receive the ESLs from their collaborator, or you can go to the findmice.org, that website, and if you type your genes and or you enter several parameters, you maybe find out the mutation you want, they already exist. And that website, findmice.org, they may tell you, okay, we have some sperm available, frozen cryo sperm somewhere, or they have ESLs in some kind of repository centers. One thing for ESL source, once they come from repository center, usually that was generated by large-scale ESL mutagenesis several years ago, and the ESL quality can vary a lot. Some, I think in general, so far they have probably over 50% of these cell lines, they can go germline. And once, if you receive the ESLs from this type of resource, it's better you import more than, let's say, at least 2 clones for the same mutation. And I will inject both clones, 2 clones, separate clone, you know, hopefully at least one of them can go germline. So that's a unique character for the ESLs. And ESL has been used by a lab here at the university, and they have a very good ESL quality. And the project from their lab is highly successful, I think, especially in terms of go germline. You know, I think in the last year, they have about 10 different mutations in the ESLs, and except one, otherwise they all go germline. I do have some other lab received the ESL from repository, and I inject 2 of them and 2 clones. You know, I think most of them want germline. There are 1 or 2 probably don't go germline. You know, you can control that. So the ESL quality is very important. Okay, may I have the next? Thank you. So we do have other services. Most highly requested service is sperm cryo. Most lab, I think, want to save, you know, the space or the mouse cost. Once they done with specific mouse project, you can cryopreserve the sperm, and you can discontinue the mice clones in your lab. You know, for the sperm cryo, I asked for 2 males. They have to be proven fertile, and so they do need to show that. You know, that's a request. For the age, I asking for 8 to 26 weeks old, but it's flexible, and mice need to be healthy. That's, I think, you know, given. Otherwise, even 1-year-old male or even 2 years old male, we can do the cryo. But as you know, as the mouse age increase, the male age increase, the risk of recover the sperm through the IVF, let's say, that ability will drop. You know, but in general, if the male is healthy and the sperm is fine, and you can recover them. So that's to the IVF, in vitro fertilization. That's a major import route for introducing the mutations in your lab these days. And if you get the sperm somewhere from your collaborators, and we can do that. You know, it's relatively simple. And I also have some special request for a kind of very unique project. One time a lab asked me to isolate 2-cell mouse embryos. Recently, I have a request to do some sperm mortality studies currently there's a project called blasis complementation, and that's basically inject iPS cells into the mouse blasis. So this is a special project, and I do have that. And finally, I will say if you have some ideas, want some mutations, you can go to the findmice.org to see if that mutation already exists. And then we can probably use, frozen sperm or maybe the ESLs to do that to save your time. Or maybe you just want to use the CRISPR approach, because that's relatively fast. Okay, may I have next? Thank you. So that's a timeline about mouse project. So the timeline I count in my lab, the mouse genetic lab. So like for CRISPR approach, that's take about 2 months from the time I order the mice for your project to, I have the mice weaned ready to transfer to your lab. That's take me about 2 months. Before that, let's say you want your molecular work, let's say prepare the CRISPR reagent by Brian, that's take additional time. It's not 2 months. So that's maybe take another few weeks to 2 months. So if you consider the entire project for yourself, then you also need to consider after you receive the mice from me, you need to breed them, you need to genotype these CRISPR mice. So that's take another time, So in general, in my hand, in the MGL stage, it takes 2 months for the CRISPR. The other timeline is about the ESLs. The ESLs, as I mentioned, if you prepare the ESLs, that can take anywhere, if you receive from a facility, from other collaborators, that's, if you want make the ESLs, probably by some other outside service lab or even yourself, your own lab, let's take whatever the time. And for me, my part, I have the ESL received, I order mice, inject the ESL to the blasis, and then the mice born and ready for transfer to you, that's take 2 months. And as I mentioned here, for ESL injection, you get the chimera mice, and then you need to breed chimera mice until they go germline. Then your project can work. Once they go germline, you need breedings. So that's why it's taking much longer. The ESL project usually take much longer compared to CRISPR approach. The IVF, for me, once I had the frozen sperm, I order mice for you, and that's take 2 months in my hand. The other service is the frozen embryo recover. In the old days, they do have, even nowadays, they do frozen embryos for specific reasons. And to recover them, that's also take 2 months. And the sperm cryo take 1 day. For the IVF, the successful rate is very high, as long as your sperm is good. Once you receive sperm from outside, the sperm may be made decades ago, long time ago. The early days, the sperm cryo quality is not as good as these days. So that's, the IVF success or not depend on the sperm, and also related with mouse strains. B6 mice is relatively good. Okay. Next. Yeah, so this is the overview for the summary slide, and you can actually find it in my website, the MGL website. Here list the cost. Cost is many lab concerns. For a CRISPR project, if you ask work with the genome engineering shared resource, that cost is separate from MGL. The MGL will charge for MGL's work. If I use FAB mice, the cost will be $545 for a project. That's a regular price, and the cancer center member is half of that. Most lab asking for mutation in the B6, C57BL6 mice. So that kind of project will be $5,050. Cancer center member cost will be half. And the cost in here, I write down also including the per diem. The per diem cost basically is once I inject ESL into blastocyst, I transfer that blastocyst to a female. Then I put into a specific cage. That cage use PI's breeding card. That's where this per diem cost refer to. That's lab's responsibility. So that's probably not too much. That's basically cage card charge per day. For the ESLs per project is $2,360 for non-cancer center member. And the sperm cryo, I need to explain, if you have 1, 2, or 3 different mutant lines, the cost will be $505 for regular price. If you have 4 lines, that will cost will be double. So this price is non-cancer center member. Yeah, the cancer center member will be half price, and so you can see the cost in here. I will not explain, except there's a special service because it's special. So we kind of at this point we just charge $505 per day, plus if the special project need mice, and then the mice cost is yours. There's one thing I need to probably mention for, let's say, the CRISPR project. If you need a special mice like the NSG mice, that's a immune compromised strain. That mice is very expensive. In that case, we ask PI's lab to pay the cost of the mice. That mice can be very expensive. One female, I think, cost probably thousands of dollars, and I need relatively more, so that's more expensive. Or if you have, let's say, the NSG mice yourself, and then maybe I can use it. We have to talk about it. The next slide. Yeah, I need acknowledgement. So my supervisor is Tim. Then I have a part-time Erin. She's very helpful. Without her, I don't think I can do my work. I also need to thank Genome Engineering Shared Resource Director Eric and Brandon. Without their support, I don't think I can collaborate with Brian seamlessly to serve the community. And then I need to answer any questions if you have. Yeah.
Thank you, Yoon. That was a really informative talk. Would you recommend that when a new line is made, investigators ask you to freeze down sperm right away so they have a backup in case something happens?
Yeah, usually I didn't reminder, I haven't reminder any PI yet. Yes, I can do that. But usually, because MGL lab, just myself and also have a part-time staff, we don't do, let's say, I don't breed mice for PI, let's say, for let's say sperm cryo, So I ask the lab to prepare the male. Once the male ready, I will do the sperm cryo. That's not a problem. Yeah.
Okay. And that's stored on site in your facility?
Yes, that's stored in a liquid nitrogen tank in my facility. These days, I ask the lab to take half of the cryo with the sperm, frozen sperm, to their own lab, just for the security. In case something happening in MGL or any other facility, we can have a backup. So you have frozen sperm in your own lab.
Okay. And are the rates of success with non-B6 or non-FVB strains equivalent to those? So if you're doing BALB-C, is it harder to get the, the desired progeny that you want as it would be with B6?
Yeah, that's a great questions because that's one good thing for CRISPR. You can do it in any mouse line you want in the background. That's depend on the strain. Some strains are very good to make the transgenic mice, like the FAB embryo is good. So far, I haven't tried any other line here at the university, except, so the B6 is most common, the FAB, and also NSG mice. All these works well. So other line, I haven't tried yet, but I know some line is good, some line is not. So it has to be, I have to know exactly what kind of strain you want to use to make an evaluation. So if we should do that or maybe try that,
Okay. And then you gave timelines, which were really useful, but that's from when you start on a project. Is there much of a backlog, or I'm sure it varies over the year, but is there much of a backlog? So if I went into you tomorrow, for example, could we start right away, or would I have to wait 2 or 3 months?
Yeah, great. That one is vary. At this point, let's say, I can schedule you immediately. I have some scheduled ESL injection in March, and have, let's say, 1 sperm cryo will be done this week. Otherwise, I can schedule whatever your project now,
Okay.
But at some point, say in the past 4 or 5 years, I always have like within the 1 month, it's usually 4. The next month, usually have some space and can put your project in there.
Okay.
So that vary, yeah.
Are there any questions from the attendees? All right, I'm going to make a plug for your facility. We're incredibly fortunate to have Dr. Yu do this. He is almost a unique individual. There aren't very many people like him, obviously. And he needs help. So if there are any trainees or anybody who's interested in this technique, maybe we could talk and see if there are opportunities for you to learn and work in the MGL facility. Is that a fair comment, Dr. Yu?
Yeah, yeah.
Okay. All right, well, if there aren't any questions, I'd just like to thank Brian and Yoon again for this fantastic presentation. These resources are just phenomenal. And we're fortunate in that we have individuals that can perform these state-of-the-art technologies. And then also with Eric and Brandon in the background, if you will, the expertise is there at every level. So take advantage of it. And as you can see from the MGL prices, we're getting a 50% discount because of the our core grant. And so thank you to the Cancer Center for this. And Brian and Yoon, thanks very much. This was great. And I hope I can use you guys in the near future.
Thank you. Thank you.
Thanks, yeah.
All right. Thanks very much. Bye-bye.
Bye.